Thank you, organizers, to give me this wonderful opportunity to talk about our recent work about the our bioinformatics study for the coronavirus spike protein. Just very quick review for the coronavirus spike protein. So the coronavirus is a very big RNA families. There are 7 coronavirus could infect the humans. 3 pathogenic ones, the SARS-1, SARS-2, and also the MERS. They belong to the beta coronavirus. Their genome structure are quite similar. So they encode about like 16 non-structured proteins and also 4 structured proteins. Among all these proteins, I think the most famous one is the spike protein. So it mitigates the virus into the cell. In the SARS-CoV-2, they had 2 subunits, S1 and S2. So S1, they use the receptor binding domains that could interact with the human receptor through the receptor binding motif. So in the SARS-1 and SARS-2, they use ACE2 as a receptor. The MERS, they are different, use DPP4. Because coronavirus mutates so quickly, so it generates a lot of mutations. Most of these mutations are function neutral, but some mutation may enhance these transmissions. So that way, why we have so many variant strands like Alpha, Delta, or Omicron. So we want to know how could we predict the mutation effect. So here we use the computational saturation mutagenesis to look at the effect of every possible mutations. So basically for each amino acid in the protein, we mutate to the other 19 amino acid. Then we apply 2 approach. One is the sequence-based machine learning. We look at the mutation pathogenicity, protein stability, and also to see if this mutation involved to the post-translation modifications. And today I'm focused on talk about how we use computational biophysics to look at how this mutation affect protein stability and protein-protein interactions. I mainly talk based on our this paper, focus on the SARS-CoV-2. So first is we calculated their protein folding ion change. If this folding ion is greater than 0, that means this mutation could destabilize the protein stability. Otherwise, it could stabilize the protein stability. So we use this method to investigate about 80,000 mutations in the full-length spike proteins. And we also calculate the binding affinities. If that mutation, the trapped LG is greater than 0, that could weaken binding affinity. But in a few cases, that mutation could enhance the binding affinities. By this way, we look at about 3,700 mutations in the receptor binding domains. And also we look at some mutations in the ACE2s. Then look at the protein stability first. We found that the majority of this mutation could destabilize the protein stability. About like 31% could strongly destabilize the full-length spike, and also 30% is moderate to destabilize that. There are very similar patterns in the receptor binding domains. Then this line chart shows this. We look at all the mutation mean values at this residue, try to look for some function important site. It ranges from like G431 to the S514. So if we look at these top residues, we found that the most mutation in the G431 could decrease the protein stabilities. There are very similar patterns in the SARS-CoV-1 because the G is a very small amino acid, so any mutation could inferable for this stability change. Then we identify the S514. In the sequence is far away, but in the structure is very close. We find that most mutation in this residue could make the receptor binding domain more stable. Also, we found that there are very similar patterns in SARS-CoV-1. Then we look at the real mutation, the viral mutation. D1614G, so this probably is the first dominant variant strand. So when we calculate the delta G, we found that the G16 14 is the most stable forms compared to other possible mutations. It's also very interesting because it's residue is very close to the N-link glycosylation site. So we think this mutation may enhance the fitness of the SARS-CoV-2 through increased protein stability, or maybe also involved to the post-translation modifications. Then the second, we look at how this mutation could affect the binding affinities. We found that most of the mutations that have the very limited effect, except that mutation located in the receptor binding motif. It's very interesting, like we found in the viral mutation, about 9.3%, the light blue part, they could enhance this binding affinity, but in computational predict ones, they are only like a 3.4%. So then we take a look at the top case, the G496. So we can see in the most mutation in this residue, they could highly reduce these binding affinities. It's very similar in the SARS-CoV-1. It's very interesting in this label residue, F497. We can see many mutations could actually enhance this binding affinity in the SARS-CoV-2, but in the SARS-CoV-1, they have almost no effect for this binding affinity. So this residue could be very important for this spike AC2 interactions. Then we look at some example for the real variant mutation, L452R. So if we calculate the trouble DRG binding, it's minus 0.395. This indicates this mutation could enhance these binding affinities. Actually, very interesting, we look at this in the earlier pandemic. Then in the next year, so this mutation becomes a mark mutation for the California strands. At the LA Teng report our study. And recently, we found this mutation exists in the Omicron BA.4 at 0.5. So it indicates this trouble DRG could be very useful features for understanding the dominant variant. So because time limit, so and just interest us CO2, so but we also look at other MERS, SARS-CoV-1, and we also try to develop some drugs. And we also apply to the other target protein and use like a fruit fly to find some very interesting phenotype. So just summary, so we use this approach to look at how mutation have had protein stability and protein interactions. We identify some function site, and also we showed some viral mutation could be very interesting, could stable the protein stability or maybe enhance their binding affinities. So thank you for my group. And also, because today I introduced structured modeling, we also work on the next generation sequence and machine learning. So we are collaboration, and also have some students and postdocs to welcome to contact me.
Thanks, Shaolei. As we transition slides, I think we may have time for one question. So.
Yeah.
This? Okay. Thank you very much. I wonder what's the correlation with between the delta Gs that you calculated and, for example, Bloom's data for the RBD, for the mutations in the RBD?
Delta G with RBD.
So
could you repeat that?
What is the correlation between your delta G calculated values for the mutations in the RBD with the experimental data of Bloom's lab?
Oh, yeah, that's actually a very interesting question. We saw that I think the overall its correlation is low, but in that receptor binding domains, we found some very high correlations.